Okay, so now 12:02, so let's begin. Hello, everyone. Welcome to the Environmental Research webinar. I am Sung-Joon Park, faculty member of Epidemiology and Environmental Health Sciences. So this seminar series is sponsored by the Michigan Life Stage Environmental Exposures and Disease Center, in short, M-LEEaD. I'm very excited to have this opportunity to learn about an emerging environmental health issue that we may have been affected for many decades, but have not realized its impact. So this is plastic pollution from micro level and nanoscale plastic. So today, the M-LEEaD Integrated Health Science Core hosts this seminar to learn about the current status of science of nanoplastics. And then we will have another session next week that the M-LEEaD Community Engagement Core will host a residents and researchers discussion on plastics, policy, and public health. So please join us next week as well. So I'm going to introduce today's speaker, Dr. Matthew Campen. So Dr. Campen is a distinguished professor in the University of New Mexico College of Pharmacy. He co-directs the UNM Clinical and Translational Science Center, as well as National Institute of General Medical Science-funded Center for Matters in Biology and Medicine. Dr. Campen received his PhD from the University of North Carolina School of Public Health and trained as a postdoctoral fellow at the Johns Hopkins School of Medicine. He has studied the systemic vascular toxicity of complex, diverse inhaled pollutants for most of his career. His lab has recently begun researching the human health impacts of micro and nanoplastic as an emerging toxicant of global concern. With that said, so please join me on welcoming Dr. Campen.
All right, thanks, Sung-Kyun. I appreciate the opportunity to come and share our work. Welcome, everyone, to Albuquerque, New Mexico. It's all right here. It's not as sunny as you might hope today, but it's usually really nice. So I'll share this. Here we go. So our lab started out very skeptically in the world of microplastics and nanoplastics pollution. And now we're kind of all in with 2 feet. And I'll explain a little bit of my journey into this field and some of the discoveries we've made, including the image that you see on the right-hand side, which is the isolated nanoshards of plastic that we can pull out of human brains, decedent brains. So I started out very skeptically in this area because I've studied air pollution like diesel exhaust particles and coal combustion and all these really horrible super toxic particles, right? And plastics were often used in our field as a negative control or, like an inert particle that would mount it would generate some sort of inflammatory response at high levels, but certainly not something your body really responded to. And it wasn't until I did this project with my son in 2019, ahead of the pandemic. So this was his 7th grade science fair project that ended up not getting presented because of pandemic cancellations. But he's here in the Rio Grande, near the headwaters of the Rio Grande up in Colorado. It starts just west of Creede and it flows down south through the San Luis Valley to Taos. It then hits Albuquerque, where we live. It continues down to El Paso and then runs along the border of Mexico and the United States, although it dries out, and then it really reforms around Big Bend National Park in Texas. But Juarez and El Paso take a lot of water out of this river. Now, needless to say, it is a very important drainage system for our entire state, and it has both an agriculture and a cultural and historical relevance for our region. Each of these little asterisks, these little red crosses, these are Pueblo communities that have evolved essentially along the watershed region. So they're in that region specifically because of this river. So what we did was we sampled using glass bottles the water along three different locations, and so geography professor friend of mine estimated that only about 700 people can contribute to the watershed above this first sampling point, and then about 77,000 people contribute to this point in Taos. And then about 1 million, a little over 1 million people can contribute to the watershed leading to the south point of Albuquerque where we sampled. And what we found, using relatively simple techniques, a black light and a microscope and some filter paper, we found that this increased as you went down the river. So not surprising, increasing amounts of human impact lead to greater numbers of particles that you can identify with relatively simple techniques. But what really got me interested in this, what really grabbed me, was how easy it was at the headwaters of the Rio Grande to identify plastic particles. So this is really the global problem. We've been generating and using plastics for about 50, 70 years, thereabouts, and it's been at an exponentially increasing rate. The doubling rate of plastics generation is about every 10 to 15 years. And it takes about 30 to 40 years for those plastics to degrade into micro or nanoplastics. And so we're seeing a mirror image of the formation of micro and nanoplastics in the ocean, and probably also in the groundwater and also in the crops in the fields that we grow. But there's a delay factor. So even if we stop using plastics today, that's the generation of microplastics from plastics already in landfills is going to continue for generations of humans to come. The problem is plastics never stop being plastics, and they just get smaller and smaller and smaller. So they are exposed to UV light, heat and cold and water and chemicals, and they get smaller and smaller, eventually becoming these nano-sized fragments. There's actually very little information about what these things look like when they get past the size of about a micron. And I like this scale bar on the right-hand side to demonstrate when we talk about things larger than 1 micrometer, which is 10 to the third nanometers, 10 micrometers to 100 micrometers is about the size of the cells of your body. I think megakaryocytes in your bone marrow are about 60 microns, and those are about the largest cells you have. Whereas red blood cells are only about 7 microns, just to give you some perspective. Viruses, COVID virus was about 50 nanometers, and they're sometimes upwards of 100 nanometers, but I'm going to show you plastic fragments that are in this range, super small, and easily taken into the human body. If there's any takeaway, I want people to understand that it's not particles from fresh plastics. This is my prop bottle of water here. This bottle of water will not give me meaningful amounts of microplastics, but eventually it'll degrade and get into the environment and stay in the water system, get put onto our fields, and after 30 or 40 years of UV light and heat and cold, it'll get into the food we eat, and then we'll be exposed to it. Our first real foray into this was asking, whether the polystyrene microspheres that are easily available for research purposes would be a viable model for us. So we got these 1 and 5 micrometer polystyrene microspheres, and we thought, well, what would happen if we just aged them under UV light for 5 weeks? We were able to show that, yes, in fact, this arrow is pointing to this little blue hump, which is the increased number of carbonyl formations on the surface. That's indicating that there is an oxidative change. There was a visual change, some roughening of the surface. So we definitely had something happening. And then when we put those into cell culture and this is just a wound assay where we're looking at the regrowth of cells. The figure on the left-hand side shows the fresh, pristine polymers. We were able to see a minor reduction of regrowth in this assay, but there was a dramatic effect when we aged those particles. So, okay, that completely reset our thinking. And I'll refer you back to this play. This is in Monument Valley, Utah. This is called Forrest Gump Point. It's the scene in that movie where he decides he's going to stop running. But it's a point of inflection. And so we sat back and we thought, are we doing anything relevant whatsoever with these sort of assays? Are we using a correct dose? Are we using the right shape of material? Is the polystyrene relevant? Is the age of the particle relevant? So we decided what we needed to do was figure out what is actually inside the human body. And this has been a fun journey because we've totally thrown everything up in the air and said we don't know anything. We're approaching this from the standpoint of ignorance, and we're going to start learning as a team. And it's been great because we've got people at all levels contributing a very multidisciplinary brand of projects and expertise involved. So, first question is, are we using realistic doses? How do we study what's in humans? Well, up till the last couple years, really the last year, most of what had been identified in humans used visual microscopy using Raman or FTIR spectroscopy or LDIR spectroscopy to identify a spectroscopic fingerprint that says this is a polymer. These are really nice assays to use, but the problem is a lot of the systems don't even reveal particles less than 5 micrometers in diameter. As somebody who studied air pollution all my life, I know that PM 2.5 is the standard globally. So we don't even worry about something unless it's smaller than 2.5 micrometers. And of course, the University of Rochester made a big case, very important case about ultrafine particles, which are 100 nanometers. So we knew that we had to look at something smaller. So we developed a more cumulative approach for capturing all of the plastics independent of size and using a spectrometric method to do this. So if you're familiar with Breaking Bad, the TV show about New Mexico, this is what we do to tissues out here. We put them into either an acid or a base, and we digest everything away that is biological. Essentially, we're using potassium hydroxide, turning the tissues into soap. We then spin out the solid remaining particulates, which is, plastic and some small rocks, but mostly plastic. And there's some other biologics that are not fully digested, and I'll touch on that. We then use a process called pyrolysis GC mass spec, which takes the pellet derived from the ultracentrifuge, sets it on fire, and we look at the smoke combustion that comes out of that. So the ions that are generated in that combustion can be traced back to specific polymers. So, for instance, different peaks coming off of this in D is the chromatogram, and you can see the mass spectra from each of those. This is an example of polypropylene from a placental pellet versus the polymer standard. You can see there's tremendous amount of coherence. At another peak, we see the ions generated for polyethylene. Another one would be polystyrene. So we can look at a lot of these things all at once. And I like this quote from Marcel Proust, that we're now really looking at this problem with a fundamentally different tool. It's giving us really new insights. The first paper we published on this was Marcus Garcia's, really his first paper as a postdoc, showing that we could identify plastics of different polymers of plastic in every human placental sample we looked at. There were 62 placental samples. By and large, the average was low, around 100 micrograms per gram. But there were a lot of subjects for whom the values were 6-7 times that much. And so each of the different colors here is representing a different polymer. So polyethylene, polyvinyl chloride, nylon, PET, styrene, butadiene rubber, etc. And you can see that by and large, polyethylene is the dominant polymer that we see, followed by typically polyvinyl chloride and nylon and PET. Typically, we see more polypropylene as well, and I'll show you more from different organs where that's more prevalent. The important thing of doing this and having this method is that now we can link concentrations of plastics to health outcomes. And this is sort of our first endeavor with this, working with Enrico Barozzo from Baylor College of Medicine. And if you look down in the lower left-hand corner at B, this is looking at the cumulative amount of plastics as we measure with this assay and comparing placentas from term versus preterm. And you can see that there tends to be higher concentrations of plastics in the preterm placenta, which is impressive given that that preterm, by definition, is about 3 or 4 weeks younger than the term placentas. So there's greater amount of accumulation in those. Whether that's cause or effect, we can't say. We also see some differences between ethnicity and race in these populations, with non-Hispanic black and non-Hispanic white mothers having higher concentrations in the placenta, both at term and preterm. Okay, but the big project we had going on in the background was working with our Office of the Medical Investigator to obtain specimens from decedents. These are autopsy specimens. And we were able to get a number of different tissues from them because they hold tissues for forensic reasons. They just are legally bound to collect specimens and hold them for a time. So we were able to go back to 2016 and collect those samples before they were discarded. And we did a head-to-head comparison of liver, kidney, and brain, specifically the frontal cortex. We also were able to get more recent decedent samples from about 2024. They were technically from November 2023 until January or February of 2024. But we were able to span essentially an 8-year gap. We ran the pyrolysis GC mass spec on the pellets derived from these samples, and we were able to show two things that were very compelling. First of which is that from 2016 to 2024, by and large, the microplastics seem to be increasing in concentration. This was not the case in the kidneys, but it was in the liver and in the brain. We also noticed that the brain is about 10 to 20 times higher concentration than what we see in the liver or kidney. Added to this, the little orange dots were independently run samples at a collaborator, Jorge Gonzalez at Oklahoma State, also has pyrolysis GC capability. So we confirmed that his results were ballpark what we were seeing as well. If you're wondering about the demographics of this population, so they're about similar. I mean, we don't know a lot about these decedents, but the 2016 and 2024, mostly the age is something we should look at as being an important factor potentially. 45 years old and 51.4 years old, but a large spread in the age group. So if you look at the graph, you'll see in blue is the 2016 cohort and in orange is the 2024. One, there's a lot of overlap across the ages, and two, there's really no trend in either group for there being an age-related accumulation of plastics, which is interesting. So we think there's really an equilibrium that is hit over months to years that each individual has based on their own diet and or lifestyle, and possibly genetics. This is all in preprint right now, and we're responding to reviewer comments. And one of them was, well, you need to show us more data in at a different time point. So we were able to get East Coast samples, so geographically different. These were donated to. There's many differences with these East Coast samples here shown in blue compared to the New Mexico samples I just showed you, but you can see that by and large they are lower. There's this consistent trend here. We were also asked to look at dementia cases, and I will tell you right now that this is probably higher than they really are. We're redoing our digestions right now to more completely remove any potential biological interference in our spectra, but they were quite high, considerably higher than normal. That gets to, I just want to be frank with some of the issues related to pyrolysis GC mass spec. It's not above reproach. And there are concerns that lipids specifically can look like polyethylene. And so polyethylene is the largest amount of polymer that we see. We've been jumping through a lot of hoops to establish that no, it's not just, we're not just measuring lipids, we are in fact measuring polymers. And so the graphs on the right are kind of showing, look, it doesn't matter how effectively we reduce or efficiently we reduce the amount of tissue from the original with our potassium hydroxide digestion, you know, theoretically, the less efficient the digestion, the greater amount of lipids are there. That doesn't seem to hold true with the numbers we're generating. There's also concerns that we think we're looking at very, very old plastics, but all our standards are using fresh polymers, and that potentially means we're underestimating concentrations. And also because we're depending on ultracentrifugation to isolate pellets, we're likely losing yield with each run that we do, so nanoparticles will not be pulled down into the pellet as efficiently as larger particles. So I showed you that slope with the total nanoparticles, and the polyethylene is largely the big contributor to that. But we also see the same time-dependent slope with polypropylene, which is not vulnerable to that interference, polyvinyl chloride, which has different issues as well, but also styrene butadiene rubber seem to be going up over time in these samples. The other observation that's been interesting is we're doing this now with a single brain, and we're looking at multiple sites throughout the brain. And what we've shown so far is that there tends to be greater accumulation of these different polymers in the white matter compared to the gray. So you can see total plastics, you can see polypropylene only, you can see polyethylene represented here. All of these are showing a significant difference in their distribution throughout the brain. Okay, so that's mass spec. It doesn't tell us much about the anatomy, unfortunately, unless we can do these sort of big, large-scale digestions. So I've talked with a number of our pathologists to get a better handle on where these might be. Rama Gulapalli is an associate professor in our Department of Pathology, and he's employing a modified approach to a polarization wave microscopy technique. This is cool. It looks at these inclusions and identifies those inclusions that have a refractile property. So this isn't 100% saying these are plastic, but we have a hunch that they are plastic. And it's interesting because when we look at these, you can see these bright spots lighting up. You don't really notice them in the H&E until you do this polarization technique, but we see them largely in the glomerulus as well as along the distal tubule. This is just another beautiful image that he collected and really highlighting the unique nature of these inclusions. They're not in the blood per se, they're in the wall of the glomerulus in the epithelial tissue there. So really, really pretty cool identification technique. In the liver, we tend to see refractile inclusions, which we think are plastic, in the lipid droplets primarily, but also throughout the liver. And you can see these kind of kaleidoscope formations that are occurring within the liver. And this is just another beautiful liver image that he's taken. So again, they're throughout the liver, but they do tend to accumulate along with the lipids. This is electron microscope, scanning electron microscope showing these clusters in liver lipid droplet and using EDS spectroscopy to show that these are in fact carbon-based. So they're not titanium dioxide. They're not some other kind of metal or mineral. They are in fact carbon. The brain is really tough. The brain is interesting. We don't see those collections. We don't see those agglomerates of particles. We tend to see them as very small individual isolates, and it's actually quite difficult to find these. You'll notice there's no scale bars, but hopefully there's enough nuclei that are about 5 microns in diameter for you to sort of compare those two. Essentially, everything we see in the brain is pretty much less than 1 micron. And when we isolate the plastics from the brains and from the livers, and we do a good job of digesting them and using benzene to pull all the lipids away, we see images like this. And this is Eliane El-Hayek. She's been doing a lot of this imaging. And we see these flakes, these shards, and they're easy to find once you do the digestion. We're jumping through a lot of hoops now to prove that these are plastics, but I can say you every test we've done, it's plastic. This is a Raman spectroscopy, and you can see that the peaks that we see in black are the plastic particles, and comparing that to a library standard of high density polyethylene, there's a lot of overlap there, minus this this peak around 1600 to 1700, which is very commonly associated with the oxidative formation of carbonyls on polymers. Aaron Ederle at NIOSH and a team at R.J. Lee in Pittsburgh have been doing the manual measurements of the length and width of these. And you can see that here in the brain, you've got a median of about 170 nanometers by about 45 nanometers in diameter. When we look at this head-to-head, now we're comparing brain, liver, and kidney from 5 different subjects where everything's been manually measured, 200 particles or more per subject per organ. And you can see that there's a significantly larger particle in the brain compared to the kidney and the liver. This is an overall scatter of the 3,000 different particles we've sized, and you can see the orange is the brain, and it kind of sticks out as slightly larger, both in terms of width as well as length. Do we see anything that confirms the elevated amount of plastics in dementia? Yes, we do. So this is a case of dementia, and we're able to see more accumulation in 2 specific regions, which don't really surprise us. One is regions with a lot of inflammation, where there's a lot of immune cells. You can see these things taken up. Oops, I'm going too fast. But also in the vascular wall. Now, this mouse is funny. In the vascular wall, you can see a lot of these refractile inclusions using polarization wave microscopy. So I want to make it clear that this isn't necessarily causal. It could be that a disease characterized by impaired blood-brain barrier and poor clearance throughout the lymphatics may well be associated with higher levels of uptake and accumulation of plastics. So this is just a zoom in, just to make this a little bit animated. So you can see these are some immune cells from this brain from a dementia case, and there's a lot of stuff that those phagocytes have taken up inside them. But when you do the polarization, it's not a 100% coherence with just the off-colored, darker shaded things. I'm just going to go back and forth a little bit. So this technique is really fascinating because it does seem to be identifying inclusions that we would not have captured with a standard H&E stain. The other this is one of the things I think is the coolest characterization of these nanoparticles we've done, and that's simply to take the TEM grid and heat it to 130 degrees, which is the melting point for polyethylene. And you can see before heating and after heating, these flakes start to deform and fuse together. So there really is melting going on at this temperature, which is about as important a confirmation as we have that these really are plastics derived from the human tissues. So where are we? Well, these are fundamentally new observations that there are these nanoflakes of plastic inside the human body. When we look at the literature, there's absolutely nothing out there like this, except for this beautiful paper by Matt and Sylvia from the European Union. And they just published this a couple months ago, showing that they see specifically very similar compounds in individual muscles, C muscles, that I don't understand why they analyzed exactly 5 of these. I assume that the graduate student ate the other 7 from the dozen in the appetizer. But what they're showing is using a different mass spec technique. They use thermal desorption, proton transfer reaction mass spec. And they're still showing that polyethylene is by far the dominant polymer that they see, followed by polypropylene and a little bit of polystyrene and PET. Concentrations they're seeing in muscles are very comparable to what we are showing in human placentas and a lot of other tissues, about 100 micrograms per gram. But what was fascinating was they did a subsequent sequential filtration to look at the different sizes of particles, and what they showed was, yes, there was a fair amount of mass in some large particles, but from 20 to 200 nanometers and 200 to 400 nanometers were their largest bins for the total mass that they're seeing inside these sea creatures. So I'm going to keep it short, so there's plenty of time for discussion. But basically, these nanoplastics are everywhere. We're doing other studies that look in other tissues. We're looking in tonsils, we're looking in lungs, we're looking in carotid arteries and coronary arteries as well. And these are relatively easy to find. We published on the testes. The predominant form of plastics, they seem to be these nanoflakes or nanoshards. Polyethylene is clearly the most abundant, followed by sort of a smattering of polypropylene, PET, and polyvinyl chloride. But we still have problems with the detection, quantitation, and removal that we need to figure these things out. So there's a lot of opportunity in the near term for innovation around these issues. I think there's a lot of discussion related to the global plastics treaty and local, national level policies that could be in place to help lower these. But in the meantime, we need a lot more research in this area and a lot of information, a lot of innovation around how to accurately assess and quantify the amounts of these plastics in the human body. And I want to leave you with this thought. New Mexico is nowhere close to the ocean. We're a landlocked state. We're at 5,000 feet for the most part in altitude. And the decedents that we examined are not a product of eating a lot of seafood. They're not impacted by, the microplastics we see in the ocean. There's so much attention to ocean and waterborne microplastics. I just want to emphasize so much of what we're exposed to is just endemic to the agricultural system, and it's almost impossible, probably impossible, to avoid these exposures at this point. I'd like to point out some of the great people that do this work, including Elian L. Hayek, Marcus Garcia, Alex Neihart. They were principal in obtaining and analyzing a lot of the samples that I talked about. Jorge Gonzalez Estrella is a wonderful colleague out at Oklahoma State. He's been not just a willing partner to analyze a lot of the samples, but he also is informing us about the agricultural system as we go along and try to understand the bigger problem. And of course, Rama Gallipoli, Natalie Adolfi, and Daniel Gallego from Department of Pathology and Office of the Medical Investigator. And Aaron Ederle. If you don't know Aaron, you should. He's the editor for Particle and Fiber Tox, and fundamentally just a brilliant nanomaterial scientist. So it was important that we engage him on this to really get the details of these plastics. So I will stop here and be happy to take any questions.
Great. Yeah, thank you very much. Yeah, this is really interesting and also, the scary story about this. Yeah, the plastic pollution and nanoparticle. Okay, so for the participants, so if you have any question, please enter, put your question to the Q&A, and then we can cover your question. So while you are thinking about your question, so let me start with a couple of questions. So the most striking finding for me is that, so you look at the nanoplastic concentration in 2016 and then 2024, and so do you know? So can you elaborate a little bit more about why we see this temporary trend over this 8-year period, and the one thought I was thinking was that after pandemic and the people they change their lifestyle, and we use more the takeout food, and then so we use more like a plastic, and then we change that direction. And could it be a potential reason why we see this temporary increase?
It could be, but the more straightforward, if you hear hoofbeats, think horses, not zebras, right? The reality is the entire planet is using more and more of these plastics. Every year it goes up, and the generation of microplastics and nanoplastics isn't related to something you're eating now. This is not where they're coming from. This is a problem for my grandchildren, not for me. So the 2-liter bottle of Tab Cola that I threw out the window of my minivan back in the 1980s, that is probably what's inside my brain today, not something fresh. So the pandemic, it's hard to imagine that is a meaningful explanation for what we're seeing. And part of the reason we chose 2016 and 2024 is that we wanted to get away from those years as much as we could. And then again, going back to the smattering of data we have from the East Coast samples, they range from 1997 to 2013 with a median around 2004. They're all much lower. So we draw very, very straight lines when we do these simple regressions. It's pretty compelling that it's just what's reflected in the exposure in the environment.
Wow. So the usual, because now the more and more plastic, and then plastic typically are not degraded, and so that's why the more aged plastic accumulate over time, and then so that's why the humans and environment are affected by this aged plastic pollution. And so you expect that so this trend keep going up in the future, right?
Exactly, and that's my fear, because right now I can't pinpoint a health outcome that we know is going to be caused by microplastics. But as a toxicologist, dose makes the poison, and we're looking at 30, 40, 50 years of continued growth no matter what we do today. And we're so dependent on plastics at this point, between food transport, food safety, medical intervention, just the delivery of medicine. You can't do it without plastics, IV tubes, colostomy bags, things like that. So we're so dependent. There's no way we're just going to stop using plastics.
Yeah. Okay, so we have one question from the audience, and the Dr. Campen. So I will read, because after this talk, and then we will post this your talk to our YouTube channel, and then for those who are not attending this meeting, so they can see, they can hear what is the question. So hi, thank you, Dr. Campen, for the presentation. How can we decrease the use of plastic in the analytic chemistry research day to day? And as part of the committee, what are simple advices for applying at home with our family members?
So that's a great question. I'm gonna go back and share my first slide, just because it's hopefully this is it. So this is a picture of a pipette tip pulled from the beach at Camilla Point in the Big Island of Hawaii. So this washed up from the Pacific Ocean, and my team went out there a month ago to help with the cleanup and actually bring back these plastics so we can study them. But that's it. There's our impact in a nutshell. So try to think about glass and metal opportunities in your lab. Whether you're in analytical chemistry or molecular biology, do you have to use pipettes? Do you have to use those disposable things all the time? I'm guilty. I don't want to throw people under the bus, but we're trying our best to start. With especially with all the plastics analysis, we have to use glass vials. We have to use metal spatulas to move things around. So think about reuse and reusing. Same at home. There's a lot of mystery, or there's this mythology about the plastic cutting boards, because there's this one paper. That paper is describing particles that are like 80 or 90 micrometers. They're not going to get into your body in measurable amounts. So I don't want to discourage that. It does show, however, that we don't need plastic cutting boards, right? We just don't need them. There's wood, there's marble, there's all kinds of other things you can use. So, just think about in your own life the value of other materials, and try to stop giving into the Amazons of the world and the packaging that they provide.
Good. Okay, so we have two questions from Dr. Kelly Bakurski. So you can also see the question directly. Thank you so much for your presentation, Dr. Campen. These findings are really striking. Can you please expand on the idea about how other chemicals can adhere to the nanoplastics? What types of challenges these plastic toxicant mixtures might pose for conducting toxicology research?
So I think there's a lot of interest in the interaction between plastics and chemicals. Obviously, plastics are born with these chemicals, and there's chemicals that are added to them, and those can leach out. My hunch is that the nanoplastics we're seeing are just so old that if anything could have leached out, it would have by now. However, I have yet to prove that true or not. But that's for phthalates and bisphenols and the plasticizing chemicals. There's also the issue that these plastics are they're hydrophobic, right? If you've ever tried to clean out a Tupperware bowl that has bacon grease in it, it takes a lot of soap and a lot of hot water. So these plastics are going to go wherever lipids are, and they're going to take a corona of lipid and protein with them wherever they go. We don't think they get into our body completely naive and pristine. I think they come in whatever shape they were in when we ate them from the pig. Maybe I'm stuck on bacon this morning. But they were inside the pig, they were inside the corn, they were inside the, orange juice or whatever you're eating. And they had a specific size, shape, and potentially were agglomerated there. And maybe that didn't change much. We do suspect that they're hijacking their way in through the cholesterol and lipid uptake system of our body. They get into chylomicrons, for instance, and probably get distributed to the liver through that pathway, and then to the rest of their body through lipoprotein packaging. But we're still trying to figure this out. Just a whole lot of interesting opportunities to study those interactions with other chemicals, both exogenous and endogenous, is what I'm saying.
Okay, so we received a lot of questions right now. So next question is, this is also, I had the same question. Is it possible to measure nanoplastic level in blood or urine, so to conduct an epi study, large scale epi study?
Yeah. We're working on that, and a couple other labs are working on that. It's still, I make the analogy to my team. We're driving a Model T Ford right now. This is a good Michigan analogy, right? Someday we'll have a peppy Ford Mustang and with all the bells and whistles. But right now it's a new technique. We're still figuring out the optimal strategy. There's a group in the Netherlands that have done a couple of nice studies with blood, and we're using CSF actually. It's sort of like an odd thing to start with, but cerebral spinal fluid, and we find we can do a good digestion and get reliable data from that. Urine seems like it would be easy, but I don't know that the concentrations will be very high, so we haven't gone down that road. So answer is yeah. What I'll say is it takes about 45 minutes per sample to run these. So high throughput is a funny word to use. We do load them in a little carousel and walk away and let it go overnight. So each machine we can get through about 20 samples a day, but it's by no means like a 384 well plate. So yeah,
in just general population, so do we have a detectable level of nanoplastic in blood?
Oh, absolutely. We suspect it goes up and down with the diet. So, this is something we want to compare as fasting versus non-fasting. But yeah.
Okay, all right. So we have several other questions. So from Dr. David Aguilar, what biological biological mechanisms do you think are causing larger plastics to accumulate in the brain relative to other organs?
That's great. And it's funny to hear like larger plastics when they're still like 170 nanometers long. They're only the size of like 3 COVID molecules laid end on end, right? So they are slightly larger. We think it's because the brain is not built for clearance, whereas the kidneys and liver totally built for clearing stuff. They take stuff up, they push it out one way or another. So I think that's what's going on. I also think that because we see greater amounts in white matter, that there is a hydrophobic relationship there that does get in. I think we might be able to find these specifically in myelin sheaths. It totally makes sense to me that these would be caught and encircled and maybe do no harm there. I don't want to. They could also be a seed for multiple sclerosis and autoimmune diseases. So I think there's a number of possibilities around that clearance issue to explain that. I think it's a fantastic question, and we're hoping to find out.
Yeah, it is. Thank you. Yeah. All right. So another question. I wonder if there are any possibilities that the microbial community in our environment develops the enzymatic capability to digest plastics or microplastics.
I sure hope so. You know, every human being has a ton of plastic waste on their hands now. There's literally 8 to 10 billion tons of plastic on this earth, and half of it's already been put into landfill or discarded. So we're literally nowhere close to being able to remediate it all. So let's hope that something evolves. I do fear, one of the biggest fears I have is human cleverness. And the idea that somebody's going to engineer some E. coli that's going to digest these things more aggressively. And humans have a bad history of implementing, you know, even regional environmental changes to mitigate situations. So I just hope that whoever comes up with these ideas thinks through the sustainability concerns.
That's interesting. Okay. Another question from Dr. Stuart Batterman. Thank you for the great talk. Can you talk about the gaps in environmental surveillance and what you would like to see that can aid understanding and forecast of exposure trend and pathways to humans?
Great question. I think the biggest gap is not appreciating the agricultural pathway. It's sort of the nature of the beast. Just so many people who got interested in this early on were oceanography scientists, were ecologists. And so now we're seeing it in human tissues, and people are trying to explain it with, you know, what we see in the drinking water, which is actually quite low. And so I'm hoping that people start studying the soil, the crops, and the manure biosolids, that whole pathway that needs to be addressed. The other thing is, now that we're seeing these nanoparticles, we need to aggressively shift our thinking away from the LDIR and FTIR Raman spectroscopy methods, and do something more comprehensive. There's a lot of groups using pyrolysis GC mass spec, innovations around that to make sure that we're, you know, comprehensively, irrefutably measuring plastics. Those are needed. A lot of technology advances are needed at this point.
Yeah, we ran out of time. So the last question, so could our epigenome help us to fight the exposure to this plastic revolution?
Are we going to evolve fast enough? That's some hopeful thinking today. I don't know. I don't know. I think part of the problem is I don't know that we have any natural enzymes that could be tweaked to help us digest it. The other aspect is, I think the most likely concern that these things can cause is an autoimmune type of situation, as a seed for weird protein aggregation or something like that. Maybe in that sense, you can have some epigenetic evolution, if you will, that helps protect us. But I think first steps are find out what it does inside our body that causes things to go wrong, and then we can identify what the epigenome can do to help with it.
Thank you very much. So thank you for attending this seminar and also asking this very interesting and the great question. So thank you, Dr. Campen, for your excellent talk and also having this Q&A session with the attendants. So as I announced earlier, so next week we will continue this the microplastic and the plastic pollution with the community engagement core, inviting the community partners, and then we will talk about this plastics, policy, and public health. So please join us next week as well. So thank you, everyone. Have a wonderful day. Bye-bye.